We've broken the gems with recovery agent. Now we will perform the Dynabead cleanup.
Cleanup buffer comes in two sizes, one for the 48 reaction kit and one for the 8 reaction kit. For the 48 reaction kit, the cleanup buffer is thawed for 10 minutes at 65 degrees Celsius in a water or bead bath. Mix every 5 minutes. We verified there were no visible crystals and cooled to room temperature. For the 8 reaction kit, the cleanup buffer is thawed for 10 minutes at 65 degrees Celsius with a thermomixer. We have the Dynabeads My1 silane, which have been equilibrated to room temperature. We have reducing agent B, which we equilibrate to room temperature, and vortex. We verify there is no precipitate and spin down. We have nuclease-free water. Finally, we have a freshly prepared solution of 80% ethanol.
We're ready to prepare the Dynabeads cleanup mix.
Add the reagents in the order shown in the user guide. Calculate the volume of each reagent that you will need for your number of samples, including 10% reagent volume overage. First, we will add the cleanup buffer. Next, we will vortex the Dynabeads for at least 30 seconds to resuspend them. Vortex the Dynabeads thoroughly, immediately before adding to the mix. Aspirate the full liquid volume in the Dynabead tube with a pipette tip to verify that the beads have not settled in the bottom of the tube. If clumps are present, pipette mix to resuspend completely. Do not centrifuge before adding to the mix. We add thoroughly vortexed Dynabeads, reducing agent B, and nuclease-free water. Then we vortex mix thoroughly. We immediately add 200 microliters of Dynabeads cleanup mix to each sample using a multi-channel pipette and reagent reservoir. If the mix has been sitting for any period of time, be sure to pipette mix in the trough before adding the mix to the samples. We pipette mix 10 times with the pipette set to 200 microliters. Then incubate at room temperature for 10 minutes. Pipette mix again at approximately 5 minutes after the start of the incubation to resuspend the settled beads. Ensure that the samples are thoroughly mixed with the silane beads to achieve optimal recovery.
Next, we will prepare Elution Solution 1.
Add the reagents in the order shown in the user guide. Combine buffer EB, (10% tween 20) and reducing agent B. Vortex mix thoroughly, and then briefly spin down. After the 10-minute incubation step is complete, place the tube strips in slots 1 through 4 of a 10x magnetic separator HT in the high position until the solution clears. A white interface between the aqueous phase and recovery agent is normal. Carefully remove and discard the supernatant.
Now we will perform the ethanol washes.
Always use fresh preparations of 80% ethanol. First, add 150 microliters of 80% ethanol twice to the pellet while on the magnet for a total volume of 300 microliters. Wait for 30 seconds. Carefully remove and discard the ethanol wash. Repeat the ethanol wash with 200 microliters of 80% ethanol. Cap the tube strips and centrifuge briefly. Return them to the 10x magnetic separator HT in the low position. Remove and discard any of the remaining ethanol, and allow the samples to air dry for 1 minute. To avoid overdrying of the pellet, immediately add 36 microliters of Elution Solution 1 to the tube strips. Cap the tubes, remove all tube strips from the magnet, and centrifuge briefly. Pipette mix thoroughly with the pipette set to 30 microliters until the beads are fully resuspended, and avoid introducing air bubbles. Incubate at room temperature for 2 minutes. Place the tube strips in a 10x magnetic separator HT in the low position until the solution clears. Transfer 35 microliters of purified GEM-RT product to new tube strips.
We completed the Post GEM-RT Dynabead cleanup. Next, we will perform cDNA amplification.